Good evening, everyone. This is Shane Gebauer with BrushyMtnBeeFarm. I'm the general manager here. I'd like to thank everyone for joining us this evening, and I'd especially like to thank our panelists. We've got Dr. Dave Tarpy with North Carolina State University. Dave is one of the leaders in the industry when it comes to queen breeding. And knowing the ins and outs and assessing queen quality. So we appreciate him taking time out of his very busy schedule to be here. We also have with us from Massachusetts in the Plymouth County Beekeepers Association. We've got Fred and Bill up there. Fred is the former president of that association, and it's his inspiration that really initiated their group-wide queen breeding program. And Bill is the program director and is a liaison with between other clubs. So I want to thank everyone for joining us. The schedule for this evening, it is a 2-hour-long webinar, and we're going to try and hold true to that 2-hour mark. We're going to start off with Dave. He's going to give us an overview of some important factors when it comes to queen breeding and things to consider, and then we'll transfer up to Massachusetts, where Fred and Bill will go through what they're doing up there and talk a little bit about raising queens, breeding queens, and in particular how their club has designed and implemented a program that is really organized and run by the club. Of course, there's some key people in there, and those people, many of them are here with us this evening. And then we'll have a short period, hopefully at the end, for some question and answer specific to queen breeding. Oftentimes we get a lot of questions about other topics, but we're going to rein it in just to queen breeding and perhaps ways that you and a group of your friends or your association can get a program up and running. And so I don't want to take up any more time, and so what I'm going to do now is I'm going to transfer ownership of this presentation to Dave, and you should have control over the keyboard here in just a second, Dave, and I'll hand this over to you.
Excellent. Well, thanks so much, Shane, for the invitation. Thanks, everybody, for attending this evening. I've been looking forward to this for a really long time. As Shane said, we're going to have a lot of discussion here, and I'm going to try and keep my component to this at about a half an hour or so that Fred and Bill can really give a lot of the lessons that they've learned in getting their local club up and running. But what Shane asked for me to do is to provide a little bit of the background as far as the biology of queen rearing and the genetics behind bee breeding. Now, just to forewarn you, there's a lot of information here, and I don't want in any way to imply that one must be an expert in bee genetics in order to successfully implement something like this. As we were discussing earlier, Shane, Fred, and I were discussing that there's really kind of a long and very wide continuum of queen rearing, both in a passive and active sense, and then bee breeding, again, both in a passive and active sense. And so my intent here is to kind of give you a sense of what spans that entire continuum of very simple queen rearing to very, very advanced bee breeding, and what it really takes to select for these very special stocks. So, with the idea of trying to focus on the biology here rather than the practice, which we'll leave to Fred. Let me see if I can advance here, Shane. There we go. The thing to remember about queen rearing is that under normal circumstances, colonies repress their natural tendency to want to raise queens. So given the normal homeostasis of a colony, the things that suppress queen rearing are, first and foremost, queen pheromone, that is the queen mandibular pheromone that the resident queen herself produces. Secondarily, and what a lot of people forget, is that a secondary fecundity signal of that queen is brood pheromone. So brood pheromone is another thing that helps to suppress queen rearing. Bees won't do it all year long, or at least in most places, so it has to be the right time of year. They have to have sufficient food and sufficient resources, and they have to have those nurse bees that help provision the developing larvae with royal jelly. So, knowing that as a beekeeper, in order to raise queens, you have to remove those things that repress queen rearing. Namely, you have three absolute necessary conditions, three laws of queen rearing. One is that the colony itself cannot have a mother queen. You have to have lots of nurse bees to provision the queen larvae with sufficient food, and they have to be well fed, particularly with protein, because royal jelly is very proteinaceous food, so they need some good pollen or pollen substitute in order to successfully raise those queens. Research has suggested that it takes about 200 nurse bees to adequately provision a single queen cell. So that gives you a rough idea then of how large the colony needs to be in order to provide sufficient food to the number of queens that you're trying to raise within that colony. So as long as you remember ample, well-fed, queenless nurses, that is a sufficient colony in which you can raise your queens. Now, one optimal design, and again, we can have Fred go through this as to how they raise their queens and their cell starters, but the swarm box is kind of the epitome of a queen starter colony or a queen rearing colony, because it maximizes all of those different things. It has no mother queen. It has lots of nurse bees because it's primarily consisting all of nurse bees. That's the entire point of making up a unit like this. It has lots of food, especially if it's fed supplemental pollen, and preferably it doesn't have any open brood, so that the only individuals that will receive the nurse bee food are indeed those larvae that you're trying to raise into queens. Now, another aspect, not just for queen rearing, but the total complete package of queen production, of course, is that the queens also have to be mated. And this is an invisible factor that deals with how good a queen is, because you can't see how many times she's mated, you can't see how well she's mated. This is something that's very invisible to the beekeeper, but it's still equally important to raising queens and for bee breeding. Now, as we probably all know, we know that queens mate about a dozen times or more, and so the number of different drone fathers of the worker offspring can have a profound effect on the queen and her colony. Queens mate when they're very young, when they're about a week old or so, and they really only mate during the afternoons, and the weather conditions have to be ideal. So if there's a stretch of bad weather during this time of queen production, that can have a pretty significant effect overall on the quality of the queens if they're not able to mate sufficiently. Now, there are many different types of mating nukes that are available out there. These are some that we really like from Brushy Mountain that are two-way mating nukes. There's 2 nukes in each of these units here where you can place individual queen cells. They will emerge into those nukes. They will mate successfully from them and start laying in the 3 small cassette combs within these nukes. So this is a very facile way to mass produce a fairly large number of queens with fairly minimal resources of bees and equipment. Now, it's important when one is raising queens not to forget the drone sources. It's suggested that you want to put about 1 or 2 drone frames within each brood chamber of the colonies that are specifically designed to serve as drone sources for the local mated queens. If you want to time the sexual maturity of the queens and the drones at the same time, if you go through your drone sources and you see lots of capped drone brood, that's when you want to start grafting and start raising your queens. But again, we can let Fred and Bill cover that. Another thing that you want to make sure of from a genetic standpoint is that you don't want your drone sources to be, related to your queens. You want to avoid inbreeding, so you want to maintain good genetic diversity within and among your different drone sources. Now, as for the genetics, so that was a very brief overview of the how and the why of the biology behind queen rearing. We can distinguish that then from more advanced bee breeding techniques. Queen rearing is the propagation of queens, but to maintain good high-quality genetic stock, it takes a little bit of understanding of the genetics behind what it takes to have that stock. So honeybee genetics is just simply dealing with the heritable information that we see in our bees. So that key phrase of heritable information is very important, because anything that isn't heritable, there's no point in trying to breed for it. And we'll explain how in the next few minutes here. Now, the only thing you really need to know about genetics is this, and this is something that I got from Greg Hunt, who provided a similar slide one time, and I thought it summed it up really, really well. If you really don't want to listen to me for the next 20 minutes or so, then this is really all you really have to know, is that DNA, or the underlying genetics of any living thing, is just tied to that living thing in this way. The DNA itself makes proteins, or it codes for different proteins. Those different proteins are assembled together in such a way that it makes an individual bee, and then you have 50,000 bees that come together to make a colony. And that's what we care about as a beekeeper. So it's just this very linear progression from DNA to the colony that's important for bees and for bee breeding. So that's all you really have to know as far as what the DNA does. But how it influences the colony is what's important. And so that's what we'll concentrate on here. What we as beekeepers are concerned with is the phenotype of the colony. That is, how the colony behaves, all of the traits that it exhibits. Its honey production, its overwintering ability, its lack of defensiveness, all of those things together is known as the phenotype. Now, what's important to know about the phenotype is that it has two components to it. A genetic component, the genotype, and an environmental component. And so other things that go into it that are not because of the DNA that underlies it. And then an interaction term between those two, and I'll explain what that means. So in this particular example, here we have an environmental component, let's say temperature, and a response of an individual, let's say how much they sweat or something like that. And in this case, we have one genotype that has a high response, and then another genotype that has a low response, but it's totally independent of temperature. So what this means is that this particular phenotype is all because of the genes, the genotype, and has nothing to do with the environment. You can take the opposite extreme, where you have, again, one genotype and another genotype, and that response changes as a function of environment, but the two genotypes are pretty much responding in the same way. In this case, the phenotype is all because of environment, and it has nothing to do with genetics. All right? So those are two kind of extremes of how genetics can influence phenotype, either completely or not at all. That interaction term, that third part of phenotype, is something like this, where in one environment, say at low temperatures, the two genotypes are the same, But at a high temperature, the two genotypes are very different. And so that's an interaction between the environment and the genetics that can also cause differences in phenotype. So again, as beekeepers, what we're really interested in is the entire phenotype of that colony, and so that we can hopefully select for favorable traits in that colony. Now, there are many different ways that these different genes can be inherited from one generation to the next. Many of us are familiar with some of these, like recessive dominant characters, such as eye color, where the blue eye is recessive to the brown eyes, so on and so forth. These are kind of gene for gene traits that affect particular phenotypes, like eye color, like blood type, like hair color. I won't go through all of these for the sake of time, but suffice it to say that most traits, that is most phenotypes, are not governed by single genes. There's no gene for weight. There's no gene for height, right? What these are very complex traits that have many different genes that have additive effects. So you have these different gene loci that add up with each other to quantitatively result in a particular phenotype. These are known as quantitative traits, and they have many genes associated with them. So these quantitative traits then can really be viewed more at the phenotypic level like bell-shaped curves. So we have some members of the population that are very short. NBA members and other individuals in society are very, very tall, but most people are somewhere in the middle, right? And so these additive quantitative traits can be expressed in this bell-shaped curve of a particular phenotype. So when it comes to beekeeping, we have the exact same thing. We have some colonies that are real dogs. They don't build up in the spring. We have others that will just swarm like the dickens no matter what you do, but others, most are somewhere in between, okay? So for a given phenotypic trait, we can assess and assume that underlying quantitative genetics beneath it. So how can we use that knowledge about genetics to inform us about bee breeding? Now, what I mean by bee breeding here is that it's the systematic mating of selected individuals that produce offspring for the next generation, and specifically those that have desirable phenotypes, desirable characteristics that we want to keep. So it's this, the breeding aspect of it is the systematic mating of selected individuals that's very, very important to wind up with stock that we want. Now, what's important to go from generation to generation to generation through different breeding paradigms is that because only genetic information is inherited from one generation to the next, only heritable information can be selected for and can be part of a bee breeding program. So heritability is, a way to see what the phenotype of a particular parent is and how that relates to the phenotype of the offspring. And if you have a situation where the phenotype of the parent matches perfectly the phenotype of the offspring, such as this, then the heritability is, in essence, the slope of that line, and therefore, in this case, the heritability is 100%, which means for that particular phenotype, it's all because of genetics and it has nothing to do with the environment. Again, the opposite of that is when the slope is 0, when the phenotype of the parent has absolutely nothing resembling the phenotype of the offspring, or at least there's no pattern there, in which case that particular phenotype is completely due to environment and it has nothing to do with genetics. That trait is not heritable. For most traits, however, for most phenotypes, it's always a combination of the two, the genetics, the environment, and its combination, its interaction, so that the phenotype of the parent has some bearing on the phenotype of the offspring, but not a very large degree, in this case a heritability of 50%. So only those traits that are heritable are eligible for bee breeding programs and things that we want to try to select for. Now, to select for particular traits, it's important for many things to come together. The evaluation of individual colonies of these particular phenotypes, and importantly, to choose a subset of individuals within a population and allow only them to pass on their heritable information, pass on their genes to the next generation. Only by that you're selecting in a particular direction for a particular phenotype. If you allow everybody to interbreed, then there isn't any movement genetically, there isn't any movement towards the favorable types of genes that you want to keep in that population. So if you haven't really been following any of that, that's perfectly okay. This is really the crux of what distinguishes queen rearing from bee breeding. Kind of this is the ultimate end of that spectrum of what really any beekeeper can do, but just with a little conceptual know-how, this is how you're able to turn, how you're able to foster good genetics within your population and retain the favorable traits that you want. So here is the initial population up here that, you know, for whatever trait you want to select for. Name a trait, Shane. Good to select for.
Disease resistance.
Disease resistance, excellent trait, and I would think most of us would agree that that is a trait to select for. But it's also very complex where you have lots of genes. It's a quantitative trait, so you have a bell-shaped curve where some colonies are very susceptible to disease, others are very resistant to disease, or at least tolerant of disease. Of that bell-shaped curve, you have a mean score of how disease they are going to acquire of that particular disease. The bee breeder then selects a subset of that population. In this case, it would be the right tail of the population, that is the colonies that are most tolerant of a particular disease. You don't want the ones in the left tail that are most susceptible. You just choose those that are in the right tail, right? That's the breeding stock. Those are the ones that are going to be transmitting their heritable information to the next generation. That subpopulation has its own mean score of disease resistance. We'll call that mean S. The difference between those two means is quantified by S. So the mean between the breeding stock and the mean of the population overall. The individuals within that breeding stock are allowed to interbreed. Queens are raised, drones are raised from that same breeding stock. They intermate, and they produce a first generation, or an F1 generation. Hopefully what happens is, because of those favorable alleles in the breeding stock now completely populate that second generation, the bell-shaped curve has shifted in the direction that we want it to go, that is towards the more disease tolerant. So that shift in the mean of the second generation versus the parental generation from the parent to the F1 is quantified by the response to the selection, or R. And what quantitative genetics says is that the response, or R, in the ratio to the selective pressure, or S, approximates the heritability of that particular trait. And so beekeepers can very quantitatively select for particular stock of traits, of quantitative traits of interest that are favorable to them, and they can in essence, slowly march that bell-shaped curve over to where they want it, so that in the end, in the final population, you have a population of bees that have a much higher disease tolerance than they did to start with. That's what bee breeding is. It's very conceptually simple and very straightforward of what most people think it takes in order to result in stock that you want. Now, what is constraining here, or I should say what limits our ability to breed really with ease, is that it depends on the heritability of a particular trait. If there is very high heritability, that is, that phenotype is governed in large part by the genetics behind it, then the response to selection is very high, which means that you can select for that trait in relatively few generations and move that bell-shaped curve very rapidly. If, however, the heritability of a trait is low, let's say something like honey production, which has a huge environmental component and a very, very, very small heritability component, it doesn't mean that you can't select for it, but the response to selection is going to be smaller, so it's going to take many more generations to shift that bell-shaped curve in the same direction. Does that make sense? Similarly, you can impose a greater selection pressure on the population by increasing the mean of your breeding stock, that is, to be even more restrictive in your breeding stock. Rather than taking the top 25% of your colonies, you take the top 5%, in which case you can select for that particular trait in fewer generations because you're moving that bell-shaped curve, you're pushing it harder. Similarly, if you lower your selective pressure, that is, you're not as restrictive, it's going to take longer in order to move that bell-shaped curve and change the phenotype of the overall population. So that's pretty high-level stuff, and that's the ins and outs of what bee breeders do, which is try to change the genetic makeup of their population so that it's predominated by the favorable alleles, and you don't have all of these other alleles that are susceptible to disease or increased defensive behavior, all those other things that we don't want as beekeepers. And so what I'm hoping is that through Fred's example and others, that we can empower ourselves to select our own stock at a local level and at a small level by following this fairly simple concept of trying to move that bell-shaped curve to the direction that we want it to go. So to summarize, the genetic variation within a population leads to variation in the trait that we want to select for. It requires heritability, otherwise there's no point in trying to select for it. You want to impose directional selection pressure onto that population so that you weed out the bad genes and you leave in the good genes. And given sufficient time for that genetic change to occur, you can change that distribution of the trait in question of the population, and you're left with good high-quality stock. I believe that's all I have. Shane, if you wish to take questions now, or should we just hand it over to Fred? I believe I'm on time still.
You are absolutely on time, which I greatly appreciate. Now, I do have, I guess, I want to save the questions for the end because, frankly, since you had control over my keyboard here, I wasn't able to sort of sift through some of the questions that have been coming in. So I don't want to necessarily take time to do that right now.
Okay.
But what I am going to do is I'm going to pull up Fred's presentation here. and we'll hand it over to him. But I do have, I guess I'm gonna probably step on some toes here just a little bit in saying that one of the most important things that I think can be done with everything that you just talked about is there's a lot of, selecting and quantitative and trying to apply these selective pressures. And I just wanted to mention, this is more a comment than it is a question or anything else, that the way that you're going to be successful in doing that is through notes, note keeping,
Yes.
record keeping, because I think a lot of times people sort of fall into the trap of, well, gosh, this colony is really doing well. I'm going to breed from this one. And while that might not necessarily be a bad thing, there's really no track record, so to speak, for that colony. Maybe
Right.
you just caught it at just the right time, but if you came back in a few weeks, it wouldn't be that same colony.
And that's exactly right. I think the more careful you are in your measurement, the better. That is, the finer the ruler that you use, the better. A lot of people just rate it on a scale of 1 to 5 and say, 0 is bad, 5 is good. That's fine, but if you want to use a more sophisticated measure, if you're, say, selecting for mite resistance using a sticky board and you're actually measuring the mite loads themselves, that's a little more accurate, and that gives you a better picture of what that bell-shaped curve looks like. From that, you're better informed to select which ones you want to keep and which ones you don't. So yeah, you're right. There's no right or wrong approach in this. It's just about how detailed you want to get, and absolutely very good notes so that you can keep track of who is who over generations, because that's the only way that you're ever able to select for the favorable traits.
Right, and that's really where this gradient comes into play that we mentioned. We met earlier today, and in our conversation, we talked about how someone could just take a frame out of a hive that's got eggs and larvae and brood in it and put it in a queenless hive, and queen cells would be created by the appropriate age larva, but you're not going to necessarily end up with a very good quality queen at the end of that process. So that's one end of the spectrum. The other end of the spectrum is the very, very detailed oriented notes that ultimately are going to result in informed decisions in your queen breeding program, and you're going to end up with a better quality queen for the traits that you want to develop in a much faster time frame than that person that's just trying to take some frames out of a colony and put them into a queenless colony to get some cells.
Exactly right. Well, I look forward to hearing Fred and how they have successfully gone about and done this.
Yes. So with that, I'm going to turn it over to Fred and Bill up in Massachusetts, the Plymouth County Beekeepers Association. Fred, you should have control over the mouse and keyboard now. Can you just give a quick test to make sure that's true?
Okay. Thank you, Shane, and thank you, David. Why don't I can obviously move the mouse here, so why don't I go ahead and get started. Thanks for everyone attending. I want to go ahead and start. This photograph I have here is an electron micrograph photograph of a honeybee that won a photography award, and I have it in my presentation just as an indicator for how when bees start winning, photographs of bees start winning museum photograph contests, you know how much honeybees have come to the forefront of people's minds globally because of the plight of the honeybee and colony collapse disorder, etc. And it's really just an indicator of how the time is right for us to do whatever we can to see what we can do to help. Okay, so do I page down or?
Your down arrow should advance the slide.
Nope.
Try clicking the mouse. Sorry, folks, as we work through the.
Yeah, nothing seems to be working.
Let me just try. Forgive us as we just try and work through these Okay, if you click on that, you should have control of the mouse now. Just try clicking on that side, and if this doesn't work, then I'll have to advance for
you. It's not working. It's not working.
Okay. Sorry about that. Let me, there we go. So I'll just have to advance them for you.
All right, what we wanted to do as a club was attempt to raise our own queens, and we wanted to keep an eye on quality. And generally speaking, what we were finding within our club was we were finding members coming back every spring and wanting to purchase new packages every year because they were experiencing a lot of attrition over the winter. Their colonies were just not making it through the winter. So we wanted to find whatever we could do to help the survivability of our bees, and that was the first reason why we came together. All right, go ahead. All right, the most important thing on this screen really is the fact that I only had a couple of years experience raising queens before we started to do this. So it's really a slide that tries to make the point that you really don't need to have a tremendous amount of experience under your belt to attempt to do this. If you simply get the people involved, you immerse yourself in the techniques, and you commit yourself to learning about the various aspects of queen rearing, you can be very successful. Why don't we go? Okay. What we found was learning to raise queens was a tremendous opportunity for the entire club to learn a lot, and we also had fun, and we also ended up with bees that survived. Okay, this presentation I've made numerous times throughout the Massachusetts area, and one of the things that the approach that I take is, number one, sort of build a case for why even do queen rearing, and then secondly, I go into the approach that we took to raise our own queens, and then finally, I have an anecdotal example of the results that we experienced. So there are a lot of different reasons, but the focus really for our club was improving the quality. The cost of queens can be a lot higher than people want to pay. We found that the average cost of a varroa-sensitive hygienic, and I will go into that term a little bit more later, but the cost of an average varroa-sensitive hygienic queen was about $35 nationally. What we wanted to do was raise our own queens and make them available to our members at a reduced price. So we were charging $18 within the club and $24 outside for members outside of our club to buy our queens. And we had a very good demand. Let's go to the next slide. Normally, if you find that you're queenless and need to buy a queen, it can take a tremendous amount of time to get one to you. The other day we were just having this conversation where a person's hive swarmed and they wanted to purchase a queen, but none were available. And so one person said, well, why don't you just let them raise their own queen? Well, what you find is with a hive that swarms, it can be up to 18 days before you have a laying queen in that colony again. And 18 days of absence of a laying queen can result in up to 25,000 less bees in that colony. So 25,000 bees can contribute quite considerably to the ability of that colony to produce honey that year. Let's go to the next slide. And also the availability. We can just go on to the next slide. We also experienced some issues with defensiveness with a lot of the packaged bees that we purchased from the South. This is an issue that it can be difficult to deal with, but mostly what we wanted to be able to do was to simply requeen colonies that we found to be too defensive, and really you can find defensiveness anywhere, not necessarily just from the south. But the ability to requeen hives quickly was part of our motivation. One of the major aspects is we wanted to find ways to breed queens that would be able to deal more with the varroa mite. So a lot of members in our club were experiencing pressure within their colonies with varroa mites, and so we wanted to investigate that as well. Next slide. The other aspect was we wanted to raise queens up in our area of the United States in a hope that they would be better able to survive the weather that they were raised in. So if we were continuing to purchase queens that were raised in an area of the country that didn't have the same climate, for example, it would be difficult for us to expect for them to be able to survive those different conditions. This is actually as a result of our research. When I talk about research, I'm talking about reading Dr. Tarpy's research. But essentially what we found was many of the commercial queen rearing operations were somewhat challenged in being able to affect proper levels of fertilization in their queens. And from some of Dr. Tarpy's research, sometimes four out of 5 queens will not be sufficiently fertilized, and what that results in is queens that can be superseded well before you would expect them to be. So in some instances, we had queens that were being superseded in the first summer that we had purchased them from our packaged bee suppliers, and then also being superseded in the second summer. And this is not something that should happen. The other issue with commercial operations is their ability to keep queens rolling through their banks. And a queen bank is after you've raised the queen and you find that she's fertilized and laying, you need to put her in a queen bank in order to wait for her sale. What we found was that in many commercial operations, they wouldn't be able to leave queens within their mating nukes long enough for them to fully develop their egg-laying apparatus, and they were banking their queens too soon. If you have a smaller operation, you have the option of leaving queens in your mating nukes longer, or you have the option of selling your queens right out of the mating nuke so they never actually need to be banked. Next slide. And then finally, the real reason why we want to raise our own queens was to avoid this, and most people in beekeeping are familiar with what a dead out looks like in the spring. We wanted to have more bees that simply survive. The general approach that we wanted to take was that any small-scale queen rearing operation can have some advantages over a large-scale operation, and we used the analogy that the tomato that you grow in the tomato patch in your backyard can in many instances taste much better than the one you buy at the store, simply because you have a smaller scale operation. You can fertilize, you can water it, you can pull the weeds, etc. And when you expand an operation to be raising thousands and thousands of anything, it's more difficult to apply all that level of detail for quality. So this picture here is, that picture there is a mating yard with 54, it's a mating yard with 54 nukes in it, and it doesn't even take up that much space. So when you think in terms of, you know, how much room do you need to have in order to raise a fair amount of queens, it really isn't that much. The 3 factors we found that affected the quality of queens was how well they were fed during the period when they were larvae, and then how well the queen is mated, and then purely the underlying genetics. So I'll talk about how we went about addressing those 3 areas of quality. This slide is essentially the same as David's queen breeding slide, that the whole idea behind it is selecting the genetics that you wish to propagate throughout your apiary, and then raising many queens from that one isolated queen that has the genetics that you desire. What we found was that a lot of people were resistant to raising their own queens because they were afraid of some of these techniques, and one of which is grafting. You have to buy special tools, you have to be able to see really small things, and really what we did was we didn't have any experience. We just watched videos, we bought the tools, we dove in, and we were successful, and quite successful even in the second round of grafting, and I can go over that. Okay, there are a lot of different methods to raising queens. The method that we decided to go with was one outlined by Dr. Larry Connor. For anyone out there that would like to learn more about the Larry Connor method, there's a great video that is available right on the BrushyMtnBeeFarm resources area. And if you just go under resources and in the video section, you should find one, Backyard Queen Rearing with Dr. Larry Connor, which is exactly the video that we watched, which gave us the outline of the approach that we took. Okay, this is just a general outline of the queen rearing approach. There's 8 different steps here, and I'm going to go over each one. But essentially, you acquire a breeder, you let her lay eggs. On day 3, you select and graft. You install your grafted larvae into a starter colony, and I'll go into each one of these things in other slides. 24 hours in the starter colony, and you put them in the finisher. Then you have a quality control approach, which I'll go into, which is on day 7 and 8. You remove poor quality cells, and finally on day 14, you move the cells into your mating nukes, and then hopefully within 10 days, you have laying queens. Next slide. When it came to the breeder queen, we decided to take advantage of all the work done at Glen Apiaries by Tom Glenn, who has been breeding queens for Varroa-sensitive hygienic characteristics. And in a nutshell, you can go to the Glen Apiaries website to read more about VSH, but in a nutshell, the VSH genetic trait really is bees that are able to detect capped brood that is infected with Varroa, and one bee will uncap those infected cells, and another bee will dig that larvae out before the bees emerge, and they carry those larvae, infected larvae out of the hive, so that it dramatically reduces the Varroa pressure on that hive. He sells these at $150 delivered, and we selected a dark color queen in order to be more acclimatized to northern climates. This is the way the breeder queen comes in a box like this, and just wanted to give everyone an idea of this is the way they look when they come, when they're $150. Next slide. So within the box, there's about 50 attendants, and there's a candy disc down in the bottom, and this is how they guarantee that the queen is going to make it, and they will not ship to you unless the weather is predicted to be, they don't want any weather below 40, 45 degrees at night, because this is shipped overnight. Okay, the introduction cage is a technique recommended by Glen Apiaries. They claim the safest way to introduce a queen, and they want you to use this technique if you can, given the fact that you do have a very valuable queen. The introduction cage is provided by Glen Apiaries, and what you do is you pin the queen, the breeder queen, down on a frame that has open cells, honey cells, and some late capped brood. So here's a picture of our breeder queen that we've put in beneath this cage. The late cap brood is for those bees will be emerging in the next day or so, and they will immediately become her attendants. The open cells are for her to begin laying eggs immediately if she wants, and the honey is obviously so she can feed herself. We found in our experience that the bees were trying to get at the queen, at the breeder queen, so much so that they tunneled underneath the cage and released her within a day and a half. So we were ahead of schedule, and we had appropriate aged larvae to go ahead and graft from earlier than we thought. So here's a picture of my partner, and there's the frame we were about to put into the breeder colony. Next slide. Here's our breeder. We named our breeder queen Nancy, but we put it in a 5-frame nuke. The advantages of a 5-frame nuke is that you can better control the environment to prevent swarming. You don't want a $150 queen to be swarming on you. And secondly, when you come to graft, you don't have to go through as many frames to find appropriate aged larvae. Okay, the second part of the process is the starter colony. This is all outlined in Larry Connor's presentation if you care to download it. So the starter colony is a 5-frame nuke. It's a closed system, meaning the bees cannot fly. It draws upon the emergency response in honeybees because they find themselves queenless, and that provides them with the impetus to start any cells you might be putting in there. It's a ventilated box. Since the bees are not able to fly, you want to make sure that they do not overheat. You definitely don't want to put any brood in the box because you want all of the nurse bees that are producing royal jelly to use that for feeding the queen candidates. The only other frames you put in there besides the grafted cells are frames of honey and pollen, so they have what they need to feed the queen candidates. And then, very importantly, there's the moisture source in the box, which is a clean sponge soaked with water. That provides them with the ability to cool the box down if they need to by evaporating the water, and also provides them with the water that they need to produce the royal jelly. We found that through our experience that you want to shake up to a pound of nurse bees in the starter colony for every 10 queens that you're raising. We found that the more nurse bees, the better, but that sort of a critical mass there is a pound for every 10 queens. And you have to prepare the starter colony 4 to 6 hours ahead of time. So what we would do is we would prepare a starter colony, and then later that day meet and do the grafting. So I'll go ahead and start getting into the grafting now. Here's the picture. Okay. Here's the starter colony, and you can see that we've put a bunch of nurse bees in there. Those frames are filled with honey and pollen so that the bees have something to make the royal jelly with. Once again, it's a closed system. No bees are flying, and there's a wet sponge in the bottom of it. The grafting tools that we decided upon were the JZBZ system. This is a link to their website. You graft on day 3.5. What we found when we met on day 3 was, in many instances, we didn't find any larvae bathed in royal jelly, so we usually had to wait another half a day from the time that the egg was laid. You have to find the youngest larvae possible, but of course you're not going to be grafting eggs, so they have to be larvae, but as young as possible. And I'll show you how to judge relative age in the next slides. But one of the techniques that we found in order to ensure improved success is to keep the larvae in the same position in the queen cup as they were found in the cell. And this has to do with the side that they breathe upon. If you roll the larvae over, what you do is you prevent them from being able to breathe, and many of them will not be viable. The other issue is when you're grafting in the middle of summer, it's really hot, you can dry out the cells really quickly, so you have to graft quickly and get the queen bars in the starter colony as quickly as possible so that those nurse bees can start tending to those grafted larvae. Next slide. So here's the setup. We've got we pulled a frame from the breeder colony. It's got appropriate age larvae in it. We've got a grafting tool. We've got some JZBZ cups there that are lined up. You can see there's 15 cups on each one of those bars. There's a sort of a flashlight there for someone's that you can wear on your head. You know, whatever technique works for you. Next slide. So here's a very important picture. It gives you an idea of how to judge the relative age. Now, when you look at the center cell way to the left, you see that, of course, that's an egg. And then just to the right of that, where it says getting old, that looks like a very small larvae. But when you compare the size of that larvae to the one just to the right of it, you can see it's 2 to 3 times the size. So you need to choose the smallest larvae that you can, and your eyes will get better and better at it as the more you do it. One thing is interesting is that a larvae of the appropriate age is actually even smaller than an egg. Next slide. So when you graft, what you want to do is there's various tools to use. There's bamboo, and there's steel, metal tools, there's plastic, etc. And you just want to find the one that works right for you. We didn't find any one better than any other. It's just personal preference. But being able to graft that larvae and having it hang off the end of the tool is the way you want to do it. That way when you put it into the cup, you would just use the adhesive properties of the royal jelly to pull the larvae off the end of the tool, and just putting it in as much in the center of this cup as you could would improve the results.
And Shane, as we're scrolling through the next 4 or so slides, you'll see, like Fred had said, everybody has their own technique and everybody does it a little differently. But more importantly is it doesn't take a lot of experience. What it really takes is commitment from a couple of individuals, and they can learn very easily. The different tools different people handle, but it works out really well. And in the next slide or two, you'll see the importance of you know, the placement. So not only scooping up, but placing it back exactly where you got the from. So the next slide might show a little different procedure.
Yeah, next. Okay. So yeah, once again, we simply place the larvae in the cups, trying to get dead center and trying to lay them down in the center, in the bottom of the cup, in the same position that they were found in the cell.
And as we're going along, we had a face cloth that was with us, and each one that we put in, we would cover up the one that was completed to keep it moist, and also you'll see that we were trying to also mark what the cups were done, because they are so small you might lose track of them. And it's very important to keep in the bottom of the cell. I will say a little bit. It also correlates to the success rate of your grafting.
Okay, next slide. All right, here's our first grafting session. There are 15 cups on each one of those bars. So our very first grafting was simply 30 cups. And you can see Corey's got the wet sponge in his hand there, and we're about to place this into the starter.
You still with me, Fred? Hello? Dave, can you hear me?
Yeah, I can hear you.
It looks like we've lost Fred, unfortunately. He it's showing that he's gone offline. Hopefully he'll be back with us shortly. I guess we'll take this opportunity to look at some questions here, and see if he rejoins us.
He's probably still talking right now, not even realizing it.
I hope not. Let's see here what. Let's see if I can scroll through. I haven't had a chance to review these, so I apologize as I get in here. There's a question here that this is a pretty quick one that I can ask. There was a slide, Dave, that you had of a grid that was laid over top of a frame that had various colors sort of outlining that. Are you back, Fred?
Yes, can you hear me?
Yeah, we lost you there for a second. So we went to questions here real quick. Let me just finish asking this one, and then we'll get back to you and jump right back where we left off.
Okay.
So Dave, that grid that was laid over top of the frame of bees, what was that for?
That's a way to measure. I was talking about having a finer ruler, and that's one of them, where each one of those grids was a square inch, and those different color patterns that were on there signified 25, 50, and 75 square inches in kind of these semicircles that's very typical of brood areas. And so if one is going in and doing very accurate measures of brood area within a colony, those kind of pre-painted areas can help the counting go a little bit quicker. So that's what that particular device is used for.
All right. Well, thank you. So Fred, we'll go back to you and pick up where we left off.
Okay, Shane. This is Corey placing the queen cups in the starter. Once again, packed with nurse bees. No brood in there. Those other frames are just honey and pollen, and it's a closed box, has a ventilated bottom and a wet sponge in the bottom, and the cups stay in there for 24 hours. Next slide. Okay, now this is the finisher. The finisher are where all the cells are going to be finished, of course. It needs to be a very strong colony. It's okay if it's queen right, but we do require a queen excluder in between the queen right area of the box and where the cells are going to be finished. You place a box above a queen excluder with frames of brood, honey, and pollen. The brood is important in order to draw the nurse bees up into that box because the nurse bees are the ones that are going to finish these cells. You insert the started cells right into the center, and then the bees will finish whatever they perceive other bees have started, and that was, of course, the reason behind the starter colony. The two areas of quality control that are applied in the process are on day 7. You remove all cells capped early. It's very important because that means that you grafted a larvae that was too old. If you don't do that step, what you can find on day 14 when you're coming to put those cells into the mating nukes is some of these early emerged queens will go through and kill all the other queens in your finisher. The second quality control is on day 8, where you remove any cells that are misshapen or insufficiently filled with royal jelly, and there's a photograph coming up that will give you a better idea of what that means. This is just a picture of the finisher. We're showing the queen excluder down below. So the queen right area of this box is below that queen excluder, and then all these frames up above are there to draw the nurse bees up. So there are open larvae frames there, but also a lot of honey and pollen frames. And let's move on to the next slide. Here was the result of our first grafting. You can see that only 17 of these 30 cells have taken, so that's right around a 56% success rate. The good news is that we achieved 100% cells taken in our second grafting attempt. So we were very happy with the steep learning curve that we experienced in this process. Let's move on. Okay, so here I'm kind of summarizing. If everything is right, the bees will build the queen cells. The cells will be filled with royal jelly. The cells will be capped on day 8, not 7. And the cells will be large, long, and well built. Once again, quality control requires on day 7 to remove all the cells capped too early. Next slide. Okay, here's a photograph I was telling you about, which indicates the cells that are well built, long, large, and filled with royal jelly. One of the reasons why we wanted these JZBZ cups was because they're semi-transparent, and you can see at the bottom of the cup, which is actually at the top of this picture, you can see the white royal jelly is filling those cups. So on the 2 cells that are on the outside of this picture, these are going to be culled. These are there's no royal jelly that you can see filling the bottom of that cup, and also these cells are too small. It's an indication that these queen candidates are not well fed, and therefore we're going to take them out of the process now. Let's move on. Here is a mating nuke. This particular mating nuke is a standard size deep box that we modified to be what we call a quad. There are actually 4 mating nukes in each one of these boxes. We had a generous donation by one of our members and gave us 9 of these boxes, and we converted those 9 boxes to 36 mating nukes. And each mating nuke can take 2 frames, and each compartment, of course, is completely isolated from the other compartments, and they each have their own opening that you can see one opening here, but each compartment has an opening north, south, east, and west on this box. This is going to assist the queens with orientation and returning back to the proper nuke that they emerged from. Next slide. So the mating nukes, you're putting your queen cells in on day 14. But then you have the question of how often and when you're going to check the nukes. So what we found was if you check the nukes too much, you can find that the queens are jittery and they can abscond on you. So we later in the process developed an approach which was leave them alone for 10 days. The other great risk to mating nukes is, given that they are unnaturally small populations, is that they are susceptible to robbing anytime there is a nectar dearth, even for a few days. Your mating nukes can get robbed. We had certain mating nukes that were simply identified as nukes to be robbed, and it didn't matter what we did, they just continued to be robbed. And what we learned was, after area bees identify a particular nuke as a robbing source, we just abandoned it, because you couldn't, no matter what you did, you just couldn't keep it properly stocked with bees. So that's just a thing that we learned through our approach. Unless, of course, you have a major nectar flow, you can probably go back to using that nuke. Next slide. So with mating nukes, you have challenges with respect to having your queens return back to the original nuke. So we tried many different techniques. And then we also, with regard to the greatest challenge we found with mating nukes, is keeping them stocked with bees. We simply didn't have enough bees as part of our queen rearing initiative to stock 54 nukes. So we created a program within our club and asked our members to not necessarily donate, but just lend bees to us. So what we did was we created a program where any members, if they would lend 2 frames of bees to our queen rearing program, we would raise 3 cycles of queens in that mating nuke and then return those 2 frames to them at the end of the season with the last queen that we raised. So they would basically lend us 2 frames of bees and in return get a free queen. Here we have one of our later grafting sessions. We had 45. We grafted 45 in this session, and I think we're right around mid-30s that we were successful in inserting into the mating nukes. So this is just a photograph of day 14. We pull this frame out of the finisher. We bring it to the mating yard, and we insert one of these cells into each one of the mating nukes. Next slide. So essentially, you're just grabbing one at a time, and we place them in a mating nuke wedged between 2 top bars of the frames in the center of each mating nuke. Next slide. And that's really the process. As long as you're putting these cells in the top center, then you reduce the possibility that these can be chilled if you have a cold night, or them falling to the floor of the mating nuke. So really the success is to keep those cells up at the top and in the center. Next slide. In order to improve our attrition rate, which basically that has to do with the queen's ability to successfully return to the original mating nuke that she emerged from after her mating flight, we found creative ways to place mating nukes. This particular pattern was, we found that if you radiated mating nukes out from a center pattern, it works with the bee's ability to navigate by the sun, etc. We also placed all of our mating nukes that were 4 mating nukes within each box. We placed those in a large circle around the entire yard to help with that. And we only had somewhere in the vicinity of 19% attrition. So most people that we talk to who raise queens say if you only have 19% of your queens not make it back to their mating nuke, you're doing pretty good. Next slide. This is another technique we used when we had to line up nukes next to one another, is we alternated the openings front to back, front to back, front to back, which assists again in the bee's ability, the queen's ability to find the nuke that she emerged from. Okay, queen time, the timing is important, of course. That's why there are so many, queen calendars out there. As Dave was talking, there's 5 to 7 days before they even go on their mating flights, and then they can go on numerous mating flights, and then in some instances they don't return safely. But as I said, we were pretty lucky with our approach. So these 2 photographs are of a virgin queen and a mated queen. When it comes time for you to inspect your mating nukes, you have to have the ability to identify a virgin versus a mated queen. It helps you in knowing whether or not you should be looking for eggs yet. If you look at the queen on the left, which is a virgin, you can see by the length of her wings that they extend almost 2/3 of the way down her abdomen. So that was one of the ways that we could tell that this was most probably a virgin queen. I don't know how reliable that is, but if you look at the queen on the right, her wings are about halfway down the length of her abdomen, and really when a queen's abdomen is fully expanded and she's producing all of the eggs that she can, she literally is dragging her abdomen around, rather than the queen on the left, which is able to walk with her abdomen lifted up off of the frame. So it just helps you when you're going through all your mating nukes to know what you're looking at. Next slide. All right, on day 26, that's the day that you shoot for in terms of looking through your mating nukes to find laying queens. in this picture here, Corey is not wearing a veil, which some people might think it's not wise to do that, but what we found was inevitably you're going to have difficulty finding eggs and looking for queens, etc., and we had more success by taking our veils off. If anyone felt uncomfortable doing that, we just have them step 12 to 15 feet away from the mating nukes, and even though we have 54 mating nukes in our yard, they're all very small colonies, and the defensiveness was very low. So we didn't really experience a lot of stinging, even though there's a lot of bees flying in the air. When you do find your queen and she's laying and she's ready for sale, we decided that we were going to mark all of our queens. There's a lot of debate about the merits of marking queens, and some might say that it creates a foreign scent on your queen, etc. We looked at all that and we made the decision we wanted to mark our queens because it's the only way you can tell what you're doing is successful or not. The following year, if you haven't marked your queens, you have no idea if your queens are being superseded or not. So we made this decision. There are a lot of different techniques to marking queens. We tried them all. We got queen muffs. We got all the tools, the plunger tubes, the push down cages, etc. What we found was the most efficient way to mark a queen was to just learn how to grab them right off of the frame with your hand, transferring them over. Once you grab them with one hand, transfer over to your other hand where you hold on to two of their legs, grab the pen, tiny little dot on their thorax, and then either put them back into the mating nuke or into a cage for sale or into the queen bank. Next slide. All right, the queen bank. The queen bank was just a 5-frame nuke. We took all of these JZBZ cages. These are all caged queens here. And what we did was we put a 3/8-inch dowel. We cut 3/8-inch dowel off at 1-inch sections, and they fit perfectly into these cages. And then we were able to take these cages and, using that dowel, stick it right into a bottom bar that was modified to have 3/8-inch holes in it. So we lined up all these cages, and it was not necessary for us to stock the queen bank with bees because there's so many bees flying in the air that they naturally just were attracted to these queen cages. And as long as you have a couple of frames in there of honey and pollen, then all these queens will be properly fed. There's a lot of record keeping involved in this process, and one of the most important ones is the project plan. We created a Excel spreadsheet that had smart formulas that would allow us to simply modify the formula for the grafting date, and then all the other dates within the plan would change automatically. And so when we started to have 6 or 7 various queen grafting cycles in play at the same time, you needed to have a project plan for each one of those grafting sessions because there's no way you could keep all the dates in your head. So this was important for us to create. If anyone wants ours, I'd be happy to send it to them. Next slide. All right, with regard to all the other areas that need to stay organized, what we found was, of course, the project plan for each grafting session. We had a record of all the frames that were lent to our queen rearing initiative from the membership. Once again, 2 frames were lent in return for a free queen at the end of the season. We had to keep a record of where those frames were in our mating yard. We also generated a waiting list for queens so that selling them could happen very efficiently and we wouldn't need to leave them in the queen bank for very long. Of course, we kept a record of our revenue and expenses for the club. And then we also needed to keep a status of all the mating nukes in the yard. We originally tried keeping a status of the mating nukes using a spreadsheet, but since we needed to update it so much, we decided to just use bricks on top of the mating nukes, and the brick placed in a particular position indicated what the status was within the nuke. For example, if the nuke needed a queen cell, the brick would be in a certain position. If there was a virgin queen within that nuke, it was in a different position, and so on. So we could immediately walk into the mating yard and know which nukes had had laying queens, for example, if someone showed up and wanted to buy a queen, you could just walk into the mating yard and know immediately where the mated queens were, and you could just open them up and sell the queens right out of them. So the key elements, we would say, for a successful program is that your club really needs to embrace your efforts. If you don't have the support of your club, I think you're blown out of the water from day one. We found that we needed 3 levels of involvement in the initiative. First, you needed a couple of people who really wanted to dive in and run the whole program. The next level of involvement was a core group of volunteers that would show up and inspect all the mating nukes with us, because it could take up to 40 man-hours on a Saturday morning, for example, to go through and inspect all the mating nukes. And then finally, the frames that were lent to the initiative by the membership in order to support and stock all the mating nukes was the 3rd level, I would say, of involvement of the club. And then, of course, you have to have good record keeping. And then finally, the workshops that we had 3 separate workshops on Saturday mornings at the mating yard, and we had sometimes up to 20 members would show up. We'd go all through the mating nukes, and it was a source of probably the greatest amount of learning, really, in the shortest amount of time for the club. Going through and inspecting 54 mating nukes was just tremendous. So the results our first year were over 150 queens that we raised. We raised a little money for the club. Really, the money was not the objective. Remember, we just wanted to have bees that survived. But you can't give the queens away, because you just need to. We found that if you had the price too cheap, people would come and buy them and then resell them afterwards. So you need to charge something for the queens. And then we had 3 hands-on mating nuke inspection workshops. Those were fantastic. And then the initial reports we have is very strong overwintered colonies with the queens that we raised. And that's what I'll go into the next couple of slides, which is really anecdotal evidence of what we experienced one year versus the other. This picture here is a colony that not of one of the queens that we raised. This was a colony the year prior to our decision to get into queen rearing. So this is a colony I opened up on February 8th in 2010. Once again, this is just a queen that we had purchased via a package that we would purchase every spring. So this is what that queen looked like. This is what the colony looked like, or at least the cluster size, on February 8th, 2010. So the next picture is a picture of a colony that was headed up by one of the queens that we raised. So this is on February 14th of this year. And this was the strongest colony that I've ever seen in my own apiary on February 14th. But because of this particular warm day, even though you see snow on the ground, it's a fairly warm day and it's 60 degrees. 60 degrees up against my barn, I went ahead and began pulling frames. So let's go to the next slide. This slide is the outside of frame 4. The only reason I bring that up is because that's where the queen was found. The significance of the outside of frame 4 is because the queen had already laid up the inside of 4, 5, both sides of frame 5, both sides of frame 6, and the inside of frame 7. So already she's completely laid up 3.5 frames, and she's already working on the outside of frame 4. So let's go to the next slide. Here's a picture, a close-up picture of the queen. You can see the size of her abdomen. The suggestion there is she's in full egg-laying mode, because you do find that queens' abdomens will shrink in the wintertime because they're not laying as many eggs. But already on February 14th, this queen's abdomen is fully expanded and she's well into egg-laying mode. Here's a completely covered frame. When I inspected the bottoms of these cells, you can see these bees do not want to leave this frame because it's completely covered with eggs and larvae. You can also see some capped cells there. But this isn't the only frame. You can also see below that the next frame in the box, there's several frames all like this, completely covered with bees. So once again, this is really just a frame. I'm taking a photograph here. This is my call to action as I'm making this presentation to other clubs, saying, look how populated this frame is on February 14th. Go for it. It's really a call to action, urging people to consider doing the same thing we did.
Shane, another anecdotal was, you know, as we were returning the frames to all the club members in August, my wife and I had a couple of frames left over with one of the queens, and we weren't sure what we were going to do, you know, re-queen or whatever. So we went and started a nuke in September. The second week of September with just 4 frames and one of the queens. It overwintered, and as of a month ago, it was as strong as all the other colonies that we had, the other 4 hives that successfully overwintered as well. So again, it's just anecdotal, but we've had some early reports that, you know, these queens are doing well. This year, we purchased another queen from Glen Apiaries, and one of the things we did was we took last year's queen and we flooded her colony with drone cells, and now we have 5 mating nuke sites, and we're flooding those areas with the drones from last year's queen and grafting the this year's queen. So maybe David can talk a little bit more about that on the drone pressures as well. But that's why it's more of a breeding program, not artificially inseminated queens, but we're doing our best for queens that we're looking for in this area with traits that we would like.
Well, it sounds like you're doing a great job.
Shawnee, that's all that we have. We're available for any Q&A.
All right. I need to, this is a tactic to buy myself time. I'm going to start with the questions by going back to a slide that sort of caught my eye, and I'm just, forgive me as I scroll back up here to try and find it. It's this one right here. Let me bring this one up, and this is, I'm going to ask both of you this question. Oops, if I can get that to open up in, there we go. So this is your queen bank, and one of the things that struck me as I was looking at this was this cage right here in the center that I don't see really any bees on. And this is something that I've heard over the years, and there's a lot of different answers that I've gotten from people, and that is there additional selection based on the bees' part in selecting for the quality queens that are banked? So this, if we were to apply that logic that people have given me over the years, I would say that queen right smack dab in the center would be a lesser quality queen because there aren't as many attendants trying to tend to her, whereas perhaps some of these queens right over here have a lot more attendants and therefore are a higher quality queen. So I'll first go to you guys up there in Massachusetts, Fred and Bill. Do you see any difference? Are you tracking anything like that? The quality or what the bees are telling you with the banked queens?
Well, it's a good question, and it's a natural intuition to think that the one that has the least amount of bees hanging on is the one exhibiting the pheromone that attracts the least amount of bees. But what I found was I would use that when people came to purchase queens out of the queen bank. I would take this frame out and I would say, feel free to grab the one with the most bees hanging on as an indicator of, you know, how strong her pheromones are, etc. But the truth was that every time I opened the queen bank and pulled out that frame, there would be a different one that would have less bees hanging on it. So it didn't seem to be any kind of rhyme or reason. There wasn't a consistency each time I opened the queen bank that this one cage, for example, had all the bees hanging on to it. It just changed each time I opened it. So I didn't know what to take from that.
Also, Shane, this year we're leaving the queens in their homes, the temporary homes, for another week, and we're making note of the laying patterns, the brood patterns, and whatnot, and seeing what difference that gives us, throughout the year. So we're hanging on to them another week longer than last year, and we're trying to track these kinds of things. Certainly, specifically the laying pattern, but is there a tendency to group to one queen more than the other or less than the other? And we'll share that information with you later in the year.
Shane, the other thing I wanted to mention was what we found was there was a correlation between the number of bees hanging onto a cage and the tendency for the queen to be roaming around up at the top of the cage, interacting or enabling those bees to get her substance. And if you look in that particular cage that you isolated, you can see the kind of a dark indication in the throat of that cage, which shows that the queen is not really in the upper part of the cage. She's down below in the, you know what I mean, in the neck of the.
Yeah, I can see her, and I can get my highlighter out right down in here. You can sort of see a dark, as if she's down in there.
So the nurse bees or the those worker bees that are hanging on are not able to get any substance with her down there.
Right. Dave, your thoughts?
Well, I've heard that anecdote as well, and there very well may be some good merit to it, although I don't know of any good empirical evidence that indicates in neither direction. This is a study actually that we've been meaning to do and very well might get to this year. The difficulty, though, is what you said is that the queen without the workers is of lower quality than those that have the workers. And so that asks the immediate question of exactly what is quality. This particular bioassay, that is attractiveness to workers in this particular situation and context, may not be a very good reflection of the overall colony phenotype or how good that queen would be in the colony. So this is kind of more of a proximate attractiveness type of result, which may or may not have a similar result in a full-fledged colony. So those are the things that I think need to be worked out. That is, do we assume that attractiveness equals quality, or is it just a different measure of what we're looking for in queen quality, that is longevity, laying pattern, lack of swarming, carrying the genetics that we want. So those are, in some ways, kind of different questions.
All right. I'm wondering if, Fred, you can elaborate a little bit on the equipment and also the different jobs. You mentioned there were 3 levels of involvement. You mentioned that you had members contribute. Frames or loan, as you mentioned, frames to the mating nukes, etc. But did the club buy other equipment like the grafting needles? How did that all come about? And can you give us more information about the different jobs? Because you had a really good division of labor, it sounds like, and I'm sure people would like to know a little bit more about that.
Right. Well, yes, absolutely. First of all, we not negotiated, but we really had approved with the club to get a budget. And so we had a certain budget that allowed us to buy a lot of the incidentals, all the JZBZ cages, all the queen cups, and you can go through a lot of cups, and what we did find, oh, by the way, is you can't reuse them. The only way to reuse a cup is to boil it and to get all of the wax residue off of it before the bees will accept it again. If you ever try to reuse a cup, bees ignore it. So we did have, we did purchase, I think, 2 or 3 packages of bees to support the mating yard. So even though you get your membership to donate frames to stock the mating nukes, you still have mating nukes that will either increase or decrease their population over the course of the summer, and so you have to add some here, take some away there. So we had 3 colonies, 2 or 3 colonies that we used to balance the populations in the mating nukes over time. We, in terms of the various jobs, we had a certain member who was in charge of the breeder colony, the starter colony, and the finisher, all in their yard. So they would, for example, schedule a grafting session, and 4 to 6 hours prior to that grafting session, they would create the starter colony. So by the time we arrived and did our grafting, that we were able to immediately insert those frames right into the starter. We had a person that was in charge of the mating yard. That was myself. So there's a lot of mating nuke maintenance that needs to take place. We had someone who was keeping track of all the frames that were donated to the operation, where those frames were, and we also had people that donated full 5-frame nukes. So they had 5-frame nukes at home. They put some bees in them, and they would let us use that as one of the mating nukes for our operation. And then when it came time for them to get their queen, we simply returned the whole mating nuke with the queen in it to them. We also had people keeping track of the sales. So we had, you know, this, and then we also had an expense side as well, so all the purchasing of the, you know, the breeder queen, all the equipment, you know, and we decided that we were going to be drawing upon the generosity of a lot of the volunteers. Were donating various boxes that we needed to use, etc., in the hopes that the second and the third year of our operation we could continue to work within our budget and just continue to purchase more equipment, which is what we did this year. You know, we have a lot more mating nukes. We were able to purchase 5 colonies of these to do mating nuke maintenance, for example, and we had additional mating nukes that were donated to the program, etc. So that's how we address that.
Okay.
One of the differences between last year and this year is like Fred said, we purchased 5 colonies this year that we're setting up because, as Fred found out last year, we were finding robbing was going on. Sometimes the bees would leave, and replenishing them over the entire summer, we'd have to take from the host colony, if you will, and shake more nurse bees in. So that was the one thing that we did add this year that wasn't last year.
You know, also, Shane, I just wanted to mention something I failed to mention earlier, which I thought was pretty important, because I get this question whenever I make this presentation at area clubs, etc., is that the very strong colony that I have pictures of that I opened up on February 14th, it's important for people to understand that that's completely untreated. There are no varroa treatments. There are no sugar shakes. There are no pollen patties. I'm not using any sugar water whatsoever. That's 100% treatment-free colony that I simply had headed up by a queen, by a QR, one of our queens that we raised, and I just changed that queen in the fall, and that colony made it through the winter without any artificial feeding and without any treatments whatsoever. And
you know, that brings up a really important point that touches on the kind of conceptual theory behind it that I was talking about. If you want to be selecting for a particular trait, let's say mite resistance, right? And you have that bell-shaped curve in your population, but then all of a sudden you treat them, right? Your measurement of the mites, therefore, is no longer a good way to measure the differences in the phenotypes among your bees, and therefore your selection is not, you're not going to be choosing the appropriate breeder stock in order to move that bell-shaped curve towards where you want it, right? So that's really important that you need to take good notes, and you need to know exactly what you've done with your colonies so that your measurement of them is as accurate of the true biological interaction between the genotype and the environment as possible, right? So that you're not somehow biasing your sampling or your measurement of those colonies, and that's the only way that you're ever going to effectively breed towards the type of bees that you want.
I've got follow-ups to both those points. You guys are going too fast for me. I've got too many good questions coming in. So let me first back up to Fred. And you mentioned that colony and that we had pictures of. One of the questions coming in is, do you guys have any sort of follow-up program or surveying that's taking place of the people that acquired queens from your program to see how those colonies are doing? Because, you know, not to say that that colony isn't doing well because of the queen, but it could have been just by chance alone. I mean, I had a few colonies that came through like that, but there wasn't anything special about those queens. So it's anecdotal and interesting, but is there any more extensive work being done to sort of see if the queens at large are also showing similar trends?
Right, no, that's a great question, and I was hopefully very clear with the fact that our results were not very scientific. It's completely anecdotal, and what we wanted to do in the coming years, or at least we're going to start this year, is start tracking our sales. Well, of course, we tracked our sales, but the prospect of contacting all of the people that we sold queens to was somewhat time prohibitive with the way to contact them, et cetera, and you know, there's a lot of phone calls involved. We were really pushing the limits of the volunteerism capacity of our club with everything we were doing, so we really didn't have someone available to do that, but we're hoping to do that this year.
Okay. Dave, I'd like to go to you for a second. We've got a lot of questions coming in about selection and traits and things like that. And I realize that everyone's going to want a slightly different suite of traits that they're going to select for, and perhaps prioritize them differently. But from where you sit, your perspective, what would you consider to be some of the more important things that a person ought to look for when making selections for breeders?
Well, the different traits of interest depend on each individual's desire in beekeeping. Is it for honey production, or is it for disease resistance, or is it for propolis collection? You know, I mean, it really, the answer to that question is inherently an individual preference. What I would say, though, is that the more that you pile on, the heritability of all of those things, all of the different genes that go into all of these different complex traits, makes it longer, makes the heritability smaller collectively, and so moving that bell-shaped curve substantially in the direction you want it is going to take longer, right? It can still be done, but you know, that the more traits that you pile on any sort of selection regime, it's going to make it go slower, right? That's why many of the breeding programs can very easily select for something like hygienic behavior, which is controlled by, I think the latest genetic work suggests about 7 different major genes that control for hygienic behavior. all things considered, that's fairly simple underlying genetics, and so it can be selected for much more rapidly than, say, something for overwintering behavior, or overwintering capability, which, is much more complex genetically, right? So I think that the individual choice for these different breeding programs is up to the individual group or an individual beekeeper who wishes to select for them. I will say one other thing, however, and that is what initiatives such as what Fred is doing up in Massachusetts, initiative, the Born and Bred in North Carolina initiative that we've been working, trying to get local micro breeders up and running, is to try and have different beekeepers selecting for different things. If we're all selecting for the exact same thing, then there's really not a lot of a sufficient diversity out there. What we need are beekeepers in different areas of the country having their own closed breeding populations to maintain diversity among populations rather than just having one big large genetic population from coast to coast. I think that type of situation has been shown to be ripe for disease sweeps and other genetic bottlenecks that can cause other problems. And so I see these types of initiatives as being very exciting to be maintaining genetic diversity among different populations that we can tap into at the local level.
Okay. There's a rather simple question here that probably doesn't have a very simple answer. But Fred, this was sort of your brainchild, as I understand it. What did it take to sort of sell this to your club, and I guess how has it been received? And I'm sure that none of your club members are listening, so feel free to be candid.
Bribe honey, right?
Actually, it was a suggestion of one of the members of our club at one of our meetings, and what we found was it wasn't necessary really to sell the idea to the entire club, but really just a core group of people that were going to be involved in doing it. And once we got, I remember the first meeting that we called really was a meeting to bring everyone together who wanted to participate, and we watched Dr. Larry Connor's video. And I said, all we need to do now, if we decide to move forward with this, is go to our regular membership meeting and convince the club that we would just need, several $100s, $300-400 for the basic items that we needed to move forward. But essentially, we were going to promise the club that this was going to be an initiative that stood on its own financial legs. Like, in other words, we were economically accretive to the club. In other words, we sold more queens and made money for the club beyond the expense of the project. And oh, by the way, now we have queens, now we have bees that survive. So really, the benefit to the club was bees that survive, but essentially we didn't want it to cost anything, otherwise we would need that hurdle. So it's a way, the easy way to convince the club is, we're going to help your bees survive, but really, it's not going to cost the club any money. In fact, we might even make a little bit.
So any sort of dissenters, I assume, are probably on board with you.
They bought queens, too.
One question I've got here is, what percentage of sales were to club members versus people outside? And you, there's also another question here, which I think we already know the answer to, but you don't have to be a club member to purchase a queen, correct?
No, we wanted to make the queens available to anyone, and of course, when you've got queens in your bank, you just want to sell them. Mm-hmm. So what we did was, because the club really provided the financial support for us to kick off the process. We wanted the club to benefit financially, and that's why we sold them at $18 to club members and just $24 to outside. And what we did was we sent information to area clubs, other county clubs, to put in their newsletters. And then we started to find people contacting us from the outside, asking us if we had any queens to sell, etc. And we also had people come from outside of Massachusetts. I had a couple people come from New Hampshire and Rhode Island as well. So I was pretty impressed with that. The word was getting out. But also, one of the presentations that I made at an area club, unbeknownst to me, one of the purchasers of our queen was sitting right in the audience, and he was able to chime in at the opportune moment to let everyone know that, oh, by the way, I bought one of these, and you know, he's experiencing a lot of success with overwintering, etc. So there was a satisfied customer right there in the audience, and that really helped a lot.
It actually might be to our own detriment a little bit this year, Shane, is currently we have a waiting list of our own club members of 76 queens, which we're in the process of doing, and we'll be actually holding another grafting tomorrow, and we have 40 some odd queens ready to be evaluated, I guess, this week. So.
All right.
The other thing to remember is we feel like 70% of the demand for your queens really takes place at the end of the season. Everybody's looking for queens really going in the fall. So if we're having 76 queen waiting list right now at the beginning to the first half of the season, we're going to probably have triple that demand later on in the summer.
Okay. Dave, I'm going to just read this question verbatim. And then I guess I'll see if my interpretation is correct. Is there a simple way for folks at club levels to institute multivariate analyses that could be replicated and coordinated by someone like yourself to cross many geographic regions to test for optimal queens by region and to support the hypotheses that local bees are best? So in other words, we make this claim, as I understand this question, we make this claim that there is great advantage to locally reared queens that are adapted over time to their microclimates rather than queens from south going up to the north, etc. Is there any data to actually support that? And I guess this question is really, is there a way to submit data to see if that is in fact true?
I submit to you that whoever wrote that question needs to sit on a USDA grant panel when you submit these grants, because I have been unsuccessful in convincing the powers that be that common garden experiments and transplant experiments to prove just that point is vital to any further breeding programs of locally selected stocks. That just because bees are in a closed population in a particular area doesn't mean that they're locally adapted. They might do really well in a particular area, but that doesn't mean that they do better there than anywhere else, right? That's a subtle distinction, but an important one. And if you can show genetic differences in that population versus others, then you've shown actual genetic adaptation. But that hasn't been done here because we have this huge metapopulation where we're shipping genetics all over the country. And so we don't have, in our managed bee population, these kind of pockets of kind of locally adapted stock. Now, there may be in the feral population, and that's something that we are actively investigating, but that transplant type of experiment is a way to do it. It's a very large undertaking, but I think it's something that is really necessary if we're going to tap in to these potentially useful alternate reservoirs of genetic diversity.
Okay. here's a question about mite counts, and is that really a good measure of a colony's ability to handle um, disease and resistance?
Not really.
And so
Not
how might you measure that? What would be the best method?
I think that's the a better question left to Jeff Harris and all of the excellent real bee breeders down at the USDA lab in Baton Rouge. You know, they go through a very strict and regimented type of bioassay where they sample the brood, they sample the adults, they go through a much more accurate means than a sticky board. The sticky board is just the incidental dropping of dead or groomed mites, and it has a big environmental component to it. Case in point, your mite drop is going to be larger in larger colonies, right? So that in of itself is not the best measure, but it's better than none, and it's more accurate than just taking a look at the bees and guessing whether or not they have a lot of mites or not. So, you know, the entire process hinges very much on, you know, how accurate one wants to be in that measurement and elucidating that bell-shaped curve. You can use very crude measurements, which gives you an idea, but not a great clarification of that bell-shaped curve, which might slow things, make it slower than it might otherwise be. Or you can use very accurate measures and have a better idea. But what is important is not as resolute the measurement tool. It's following that design and doing it in the first place. Getting together with your local group or other similarly minded beekeepers in your area and just simply doing it. And I think that what's going up in Plymouth is testimony to how this type of thing can be done so successfully.
All right. We're getting close to the end. We've actually reached our 9 o'clock mark. I just put Fred's contact information up there because there were some questions on availability of queens, and I'm sure you're going to get some follow-up to this, Fred. In closing, I'd like to ask each of you just one last question before we go. Fred and Bill up there in Massachusetts, you've obviously done a wonderful job implementing this program and developing it and improving on it. Give us just a couple quick bullet points on some of the most important things that a club or group of individuals should consider if they want to develop a plan or program similar to this.
I think Fred touched on one of them. Is you need a couple of really committed, very energetic, devoted people to drive the whole thing. Without a couple of people doing this, you know, everybody can say they're going to help, and you know, donation of the frames are great, but it's a couple of 3, 4 people that need to pull it off, and lots of bees to help support it.
Yeah, I would say really the key was for Corey and myself having totally committed ourselves to making it happen. But we received a tremendous amount of assistance, and like I said, when we needed to go through the mating yard and inspect all the nukes, when we had a dozen people show up and we were able to execute 40 man-hours of work in a 3.5-hour period on a Saturday morning. If it wasn't for them, there's no way we could have done this. But at the core, you know, when it came to the grafting and the starter colonies, et cetera, managing all of the project plans, et cetera, it was really 2 members of the club that fully immersed themselves in the process. And I would say without a couple of people, you're probably, like, one person probably isn't going to be able to do it by themselves. And as soon as you start distributing the tasks out to too many, you sort of lose a little bit of the uniformity. and consistency of the quality of the effort.
And Shane, I think one of the unique things was Fred and Corey were able to spearhead this last year, and we've transitioned, and Bob Hickey and Fred Carlson are handling it, and I enjoy going over there working with them this year. So it can transition fairly easy, not seamlessly, but again, it's the devotion of a few individuals that is the important thing.
All right, and Dave, from your perspective, if a group of individuals, whether it be a club or just some friends, want to try and do this, what important factors would you suggest they consider?
Sit down, come to a consensus of the phenotypes that you wish to select. Don't be overreaching. Don't set your goals at 300-pound honey yields, never swarm, and 0 mite loads. You know, be biologically rational about that. Set out a plan of attack as far as how to measure your colonies objectively so that you can tease apart the genetic component from the environmental component, and just stick with it. And over time, your patience and your hard work will be rewarded. And I think that Bill and Fred's efforts and many others that are ongoing across the nation are really testimony to just that.
Yeah, I think it's worth mentioning that there are other associations out there. There are not a whole lot of them, but there are a handful that are doing this sort of thing, and so there are different models out there, some slightly different than that presented here this evening, and so there is an opportunity to take bits and pieces from them as well and learn from them. We've hit our gone beyond actually our 9 o'clock mark, and so I'd like to just thank our panelists this evening. Dave with NC State, who is tremendously busy doing a lot of great research there. Fred and Bill with Plymouth County Beekeepers Association for joining us and presenting their program that they've developed up there. I think it'd be safe to say that we're here this evening because we think that this is a worthwhile endeavor for people to undertake, and it certainly, while doing it as a group, presents unique challenges, but it can also create excellent opportunities that would not be afforded to someone doing it by themselves. And so we would encourage you, I would encourage you to take this information, go back to your associations, sit down and have coffee with some friends, whoever it is, and really try and think about this and maybe develop a program on your own using some of this information. This presentation will be available free of charge up on Brushy Mountain's website. I want to thank Fred for mentioning that URL to Larry's presentation. I did not pay him for that, by the way. That was just on his own accord, but I thank him for that. And this will be located in that same area, the video library in our resource section. It'll take me just a day or two to get it up there, but it will be there. And if you have any other questions, feel free to follow up with any of us here this evening. And once again, I'd just like to thank you all for coming and thank Dave, Fred, and Bill for their valuable time in presenting their wonderful information this evening. Thank you.
Great.
Thank
you, Shane.
Thanks, Shane. We had a blast.
Well, great. And we powered through some technological hurdles there, but got through it, and I appreciate everyone's patience in that regard. Thank you very much, everyone, and we look forward to seeing you next time. Have a good evening.
Wow, this was so